When cells play the game of chromosomes, they can senesce or divide. Under the influence of mitogens or lack of anti-growth nutrients, the cell enters cell division, where the cell cycle must be completed correctly, otherwise its fate is apoptosis. During cell division, helicase underwinds the double-stranded DNA, and DNA is synthesized in the 5' to 3' direction with the aid of primers that are attached to the 5' end. Once replication reaches the end of the lagging strand, the primer is unable to bind the 5' end. Therefore, the end segment of DNA cannot be copied, resulting in chromosomal shortening. To protect the coding portion of DNA from degradation, the end of chromosomes are capped by telomeres, which are TT-A-GGG base pair repeats of non-coding DNA. After subsequent DNA replications, telomeres shorten to the point where the cell must undergo irreversible cell cycle arrest. This is called senescence, as the cell is no longer able to divide without the loss of genetic material. Telomeres also serve other functions, such as positioning the chromosomes in the nucleus and preventing the response mechanism to DNA damage, which could trigger apoptosis. The telomerase is an enzyme that is increasingly active in highly proliferative cells, such as stem cells, germline and cancer cells, that all must maintain a telomere length in order for the cells to continuously divide. Telomerase is a ribonucleoprotein with reverse transcriptase activity that catalyzes the extension or lengthening of telomeres. The telomerase recognizes and binds to the end of telomere sequence. Using the reverse transcriptase subunit and RNA template, telomerase moves along the parental strand and reverse transcribes elements in the 5' to 3' direction. This extends the telomeres to cease or delay its degradation. Once telomere length is maintained and does not continue to shorten to the point where the cell enters senescence, the cells will continue to divide. There are several key indicators to a cell changing its characteristics and becoming cancerous. One such indicator of cancer is immortalization, or the capacity of the cell to endlessly replicate. Normal cells will divide for a limited number of times. However, if the telomere lengths are maintained, the cells will continuously divide. There are 3 necessary changes for a cell to transform into a cancerous cell. 1. The telomere length is maintained. 2. There is inactivation of the tumour suppressor genes. And 3. There is an activation of oncogenes. The first necessary change is to maintain telomere length. In cancer cells, there is an increased activity of telomerase to prevent cancer cells from entering senescence. The second change is the inactivation of tumour suppressor genes P53 and RB. Mutations in these genes stop the cell's normal response to DNA damage, which is apoptosis. The third change is the activation of oncogenes, such as RAS, which is essential in regulating the signaling of cell growth, proliferation and differentiation. By aiming to decrease the activity of telomerase and therefore stopping the immortalization of mutated or cancerous cells, is now being looked at as a possible treatment for cancer.